Welcome to all. Today we're going to discuss about soil sample collection in agriculture field. This class will follows as, as such a way. First we will discuss some introduction followed by what is the need of collection of soil samples and what are the different errors in soil chemical analysis and what is the meaning of composite soil sample and what are the tools required for soil sample and how to collect the soil surface soil samples. And after a collection of samples for plantation crops or problem soils. And finally, we'll go for processing of soil sample and collection of undisturbed soil sample. So in this manner, we're going to discuss about the soils. In this manner, we are going to discuss all these 9 topics in this video. Followed by the introduction. Soil is a very large variety. For example, if you take a particular agriculture field, you take agricultural field, the fertility status of in this corner and here or here is so heterogeneous, very diversified, and it's very challenging for getting that sample. So to overcome or to bring that to overcome that challenge, we have to follow the 2 steps. One is we have to collect the soil from a homogeneous unit, followed by and we have to process that collected soil sample by different methods. Like we have to dry the sample and we have to grind the sample and we have to save the sample and followed by mixing and partitioning and storing. So we are going to discuss about all this process in the coming slides. Followed by what is the need of collection of soil samples. So what is the purpose? Very first, to understand the fertility status. So we need a detailed thought. What is the status of fertility? What is the purpose? What is the status of nitrogen? What is the status of bulk density, other many biological chemical properties we want to study. That's why to understand soil fertility status, we need to collect the soil sample. After that, we want to take some decision. After getting the fertility status information, based on that data, we're going to make a particular decision what kind of crop we can grow and what kind of crop and how much optimum yield we can get with the optimum dose of fertilizer, followed by monitoring the soil health. So as we are getting to know the information, what is the status of the nutrient status of that particular soil. So we can with that we are giving optimum dose of fertilizer. So automatically we can check the soil health. We can monitor the soil health. And finally, we will go for support sustainability. So with all this proper information, if we know the full detail about our soil sample and about the status of our agriculture field, so we can go for a proper land management strategies and balance productivity for a long-term soil health. So these are the 4 criterias, and these are the need why we are collecting soil samples. So before collecting a soil sample, we are going to learn some errors. What kind of errors we can face during the soil sample collection? Errors are a part in any part of analysis. Soil science is not exception. So very first we can see different errors. So we can divide this. We are going to face the first error is your sampling error. So this error is almost we can see in a initial stage. As we initially we are going to collect the soil sample in the field in the same soil unit. So we can, instead of collecting a 15 centimeters depth, we may collect 10 centimeters, or we may collect in a wrong location. Might be we may collect from irrigation channels or in a shady areas or nearby the pump sets. So these are the sampling errors. And what are the sub? And there is another problem called subsampling error. Subsampling error means, for example, as I told you, the agricultural field is very heterogeneous. The fertility status of every inch will be very varies. There is no uniformity. So as we want to make it a composite sample, that means we are collecting a 10, 20, 50 samples. So what will happen? There might be some subsampling error. So for example, this is an agriculture field. We are collected the sample, one sample here, and one sample here, one sample here, one sample here, one sample here. Like in a zigzag fashion, we have to collect. So we have to mix this sample properly. And moreover, our depth should be same. Almost we have to go for 15 centimeters depth. That is your plow layer. What do you say that? Sometimes, so the depth should be uniform. For example, if we cut 10 centimeters from here, again 15 centimeters, or sometimes we may not, we may collect from in this corner, or we may not mix the sample properly. At that situation, we will face some subsampling error. And another error is your analytical error. This error mostly we can see in a chemical determination. Mostly it depends mostly on your reagent preparation, and many laboratory errors we can see in this analytical error. So now we will see about the composite sample. What is composite sample? As we told that we are going to collect different samples from a very heterogeneous field, very heterogeneous. So one sample from here, one sample from here, one sample from here, one sample from here. Like that, what we are doing means we are going to collect the sample from different location, as shown like this in this, not exactly like this, for an imitation purpose. So we will collect the different samples hardly from 15 to 20 locations. And what we are doing here, we are mixing those samples and we are homogenizing. That means we are making a composite sample, different locations. We're collecting different locations in a very zigzag manner. In a zigzag manner, we will collect the soil samples, we'll mix it. So that is called the composite sample. So it is a representative of individual soil volume. So it will represent. Take for example, in a classroom, for example, not in a classroom, example, in a particular classroom, there might be some issues like they may have time issues, or they may facing some electrical issues, some any facilities. So we can't talk all the 50 students in a particular location. So we will talk with one particular student. I think you're not getting again. For example, this is your agriculture field. This is you collected the fertility soil samples from different locations, and we are making it into one homogeneous unit. That is called your composite sample. We're collecting because as it is a very diversified, your field is very diversified. So we're collecting different locations. So in fact, I want to say only one thing: composite soil sample is taken to reduce the time and the cost in the soil analysis. In a soil analysis, oh sorry, in a soil analysis, and to bring that huge volume of soil sample into a very small, to draw the some small sample. For example, in fact, this it's a composite sample, taking reduce the time and the cost in the soil analysis of different soil samples drawn this from the same volume. So I want to say in one single word, thus analysis of a composite sample is an average of different soil sample drawn from the same volume. For example, as again we are going back to our example. In this particular field, we can see we are taking a different soil samples and making that samples into one volume, one particular volume. Okay, that is your soil sample. So during taking a composite soil sample, we have to follow some particular important points. What are those? Very first, equal amount of soil should be taken from each digging or a core. Exactly equal portion we have to take, and each sample must be taken from an equal cross section. That means hard. If you want to take 15 centimeters depth, you have to take a 15 centimeters depth in all on all, every each and every area. Followed by, we have to collect those soil sample. Objective one composite sample is taken from a homogeneous soil unit. That means we have to take it from a particular homogeneous soil unit. What is this homogeneous soil unit? We will going to learn in the coming slides. If you are conducting any experimental plots, we have to take the soil sample based on your different treatments given in in that particular experimental plot. Followed by, enough core or sample should be taken from one composite sample. Hardly from 15 to 20 samples are far enough. Or usually we can take 20 to 30 cores sample irrespective of the size of the field. So based on your field, based on your heterogeneity, you have to collect minimum. We have to collect from 15 to 20 samples are far enough to get one better composite soil sample. And the core should be taken at a random direction. We have to take a core or a soil sample should be collected in a very random direction. There should not be no any chemical interaction with the soil compounds. For example, and this chemical interaction means we should not collect the soil sample where we should not. We should use a different equipment. For example, we can't use the same equipment which is used by the fertilizer supplies. For example, if you are using a kurfi for kurfi or any bag for a fertilizer, we should not use that bag for fertilizer collection. Followed by what are the tools? So what are the samples? Soil sample tools are required for collection. So those are as follows. Depending on the purpose and soil moisture status, different soil equipments are used. What are those? Tube augers. Especially we are in this session, we are going to learn about the augers. This tube auger is very suitable for the soft and moist soil, which are free from gravel and stone. It looks like this. This is the your tube auger. And followed by the screw type auger. It is used for hard or relatively dry soil as well as stony or gravelly soils, but it is unsuitable for very dry and sandy soils. This is your screw auger. Followed by post hole auger. It is useful for sampling in excessive wet like paddy fields or rice fields. So post hole auger, we can use it in a paddy field. This is your post hole auger. And finally, we see the Dutch auger. This auger is suitable for the moist and wet soil having moderately fine to fine texture, but not for the coarse textured soil, as well as dry soils too. These are the difference, and this is your Dutch auger. And spade and kurfi. This spade and kurfi we can use for the surface soil sample collection, mostly suitable for soft and moist soil. Mostly this is, I think everyone knows about this. So these are the different auger sampling, which are used for the soil sample collection. Followed by the now we are going to see the process how the soil samples are collected. Very first collection of soil sample for evaluation from the farmer fields. How we are going to evaluate the fertility from the farmer fields. We need some equipments for collecting soil samples from the farmer's field. What are those? First, we need a spade, followed by kurfi, and we need a polythene bag which with a capacity of 1 kg, and we need a big polythene sheet, markers, and we need a paper tag for making a label. Of course, we need a pen, and we need a cloth bag. In some for the either we can use the polythene bag or cloth bag, but cloth bag is very comfortable and safe. So these are the equipments and materials required for the collection of soil sample from a farmer's field. And what is a procedure? Now we will see the procedure of surface soil sample collection from a farmer's field. So before starting, so whenever we are going to starting a sample collection, we want to see the location of that particular area, how the landform is distributed, what is the conditions, where the road is there, where is the irrigation channel is there. We want to take the consideration. If we have a soil map like this, we are lucky enough. So this is one of the satellite imagery. You can see here in this particular area there is each and everything. There is a road is there. This if you have this legend, there is a road. If you have this blue color thing is there, it is called your water body, and village boundary, everything is there. But in case if we have a no such type of map is absent, means we have to create, we have to traverse, we have to walk in and around the farm and to collect the information of that particular area like this. Say for example, the soil type in this location is different, and the soil type in this location is different, and in this location is different. So we want to come a conclusion, and we have to create a particular soil unit or soil boundary for the collection of soil sample in an agriculture field. Continuation to the previous slide. So we have to how we have to see the variability. In the first slide, I have told that agriculture field is highly variable. So how we have to see the variability with the help of soil texture. We can see the texture in this upland, this thing that is the upland is the medium land and is the lowland. The textural character from this layer to this layer, I mean this portion to this portion, or in this location, is varies. So based on that, we can come to some conclusion. And topography, by visually we can see this, you can consider as a highland, this might be a medium land, and this might be a lowland. And soil organic matter content, usually this depends on your soil color. Be the for seeing our soil organic matter content, we need some experience. Based on the soil color, we can distinguish which type of, what is our soil boundary for collecting of soil sample. And fertility, as indicated by the crop growths. For example, in this location the crop growth is very less, just imagine. So you can, based on this condition also, you can collect the soil sample. So like that, after collecting particular, after taking all this consideration like soil texture, topography, and soil organic matter and fertility, so we have to prepare one composite sample. We have to bring one composite sample. For example, just again, I'm giving one more example. This is your agriculture field, and imagine this is your upland, and this is your medium land, and this is your lowland. So based on your texture, you have collected some samples from here, like some 15 different locations you might collect. And this one will be your soil boundary. You will collect one sample from this location. Similar way like this here, some 15 samples you will collect and mix those things and make it into one composite sample. So one sample from here, like that from lowland, followed by the, if you have any difference, for example, if you see any salt affected portion is the problem soils is there, we can make it into another soil boundary here. So we can collect some samples. So based on the location, based on the topography, we have to decide our soil sample collection, followed by the variability. As again I'm told that, yeah, if your area is very subcontent, very diversified, reduce your area to 1 hectare and and manage your soil management. And if you found any spots, if you found any whitish encrustations, mostly white encrustations we can see in salt affected areas, or you might feel any calcium carbonate is there, means we have to collect the samples from that particular problematic area. Follow continuation. So after after defining our soil boundary, we want to collect the soil sample in a zigzag manner, just like this. So here, these white spots, your soil sample collection. So we have to collect the soil sample in a zigzag fashion. If you collect a straight way, you will miss that homogeneity and you won't get any representative soil sample of this particular field. And remember that we should not collect the soil sample near to the boundary of our soil unit. For example, see in this picture, we can say this is your soil unit. So there is almost like a 2 to 3 meters distance is there. So based by accepting the distance, we are collecting soil samples in a zigzag manner and we are not crossing our soil boundary units. Followed by, take the samples from individual spots after scrapping the surface litter. This is very important. So again, you are taking a soil sample from here. You are collecting a soil sample from here. Imagine in this spot. First, we have to clear the surface litter. There might be some dry leaves will be there, or some type of weeds will be there. We have to clear those weeds. Then we have to collect our soil samples. So followed by that, make a V notch. So after, when we decide our spot, we have to go for a V notch. We have to go for a V notch like this. We have to give a V notch with our spade, and we have to collect the soil sample and the side scraps. We have to scrap the sample and we have to keep it in our bucket, and we have for a mixing purpose. So your depth should be 15 centimeters or 6 inches from the surface. So this is the continuation. So after collecting the different locations, for example, in this spot, this is your soil map unit or soil boundary. We have collected from different locations. We have to mix this. We have to mix the collected spots with a hand or a clean piece of cloth. On a clean piece of cloth or a polythene sheet. We have to mix the soil sample for getting the homogenization. So after collecting that, we have to put the composite sample. So that is a composite sample. We have collected different soil samples from in a one particular area and we have mixed it. That is called your composite sample. After composite sample, we have to put the sample in a polythene bag and we have to keep a label by mentioning all the details here, like your farmer's name and address, date of sample collection, your plot number, land situation, whether you have collected that particular sample from highland, medium land, or lowland, any crop history, what is if we, what if you have, if there is an availability of crop history, we can go. And crop to be cultivated. What is a plan? For example, so there is a cropping pattern: paddy, pulse, paddy, like that. So if paddy is complete, if it's planning for pulses, then we have to decide our crop management or fertility management based on the requirement. And any other special information, like nearby, is there any big trees are there, any rock outcrops are there, you have to mention all those things. So after mentioning all those things, we have to tie the bag, and we have to send those sample to soil samples like this has mentioned here. After that, collection of the so now in previous slides we have discussed about the soil sample in a agriculture field, in a especially for a crops like paddy or shallow rooted crops. If I want to be too specific, mostly agronomical crops. But if you want to collect the soil sample for plantation crops where the root is very deep, like it grow up to 2 meters, we want to collect the samples from the sample collection is very important, because as the plant root grows up to 2 meters. So what are the points should be followed? Followed by, we have to dig a pit, or in a soil science terminology we used to call a profile, up to 180 centimeters. So hardly you can go around, you can go for a 2 meters also. So we have to collect a 500 grams of soil sample, scraping uniformly throughout the 15 centimeters depth. So we have to collect the surface sample of 15 centimeters. And also, if you found any salt crust, like salt encrustation is there, means that is called the or a problem soil. If visible, you have to collect the sample from that location. Followed by, after digging 1 meter, or sorry, 2 meters or 180 centimeters depth, we can collect the soil sample based on this depth wise, like 15 to 30 centimeters, or 30 to 60, 60 to 90, 90 to 20, 120 to 150, 150 to 180 centimeters depth, as follows. And if you found any hard pan, we have to take that sample separately by mentioning the thickness of that hard pan. And we have to put the samples in a cloth bag. As I told, that cloth bags are very comfortable and very safe compared to polythene bags, but that depends on choice. So by mentioning the sampling, name and address of the farmer, and depth of the soil sample. So we have to collect the soil sample in depth wise. So every depth will have a separate sample. So we have to mention in that particular bag what is the depth, and where we have collected, what is the number of that profile pit. We have to mention all those details. And after collecting, we have to send those samples to further processing and analysis. So everything is fine. Now we have learned how the soil sample should be collected from an agriculture field, that is surface soil sample, followed by the samples of profile or collection of soil samples in a plantation regions, plantation crops or horticulture crops. So what are the things we should not do, or we should not follow? Safety, what we can't take, where we can't take soil samples. Very first, we should not take soil samples near the bund areas, like this in agricultural bund areas. We should not take soil sample, sorry. And in undulating land areas, especially if you are collecting samples in a very large area where the heterogeneity, where topography is very diversified, we should not collect the undulating area, because there is a change, there is a error cause of error was very high in this area. Followed by nearby irrigation channels, no, we should not take. Why? Because sometimes irrigation water will bring the extra nutrients to the soil, so we may get errors if we collect the soil sample in that particular irrigation channels. Followed by the newly fertilized areas. For example, if you want to take a soil sample from a particular field, see that that particular farm or agriculture plot is not fertilized. That means we needn't add any fertilizer. And after that, localized depressions, and we should not collect the samples from a localized depression. And in shady areas, we should not collect the samples from under tree areas. That is not at all acceptable. So these are the areas we should not collect the soil samples. Thank you.